my screen, let me. Okay. So I think Dhananjaya asked me to share about my bio little bit for students' purpose. So I have a couple of slides.
Nice, madam. That photograph, mango collector, Totapuri, it is a Chittur
Yeah.
Photo, madam, yes.
Our orchard. Like, I don't have a big, huge orchard, but whatever is there, I like to go there.
Yes, madam.
Yeah, Chittur signature variety, right?
Yes, madam. Yes, yeah. Totapuri.
So
Totapuri, it's also called Bangalore.
Yeah, Bangalore variety.
Yes, madam.
I was talking to one of the merchants a few days ago.
Yes, madam.
He was saying that is the like what do you say? Guaranteed cash crop. The other varieties,
yes,
sometimes you may have price, sometimes no crop at all. But this is a like consistency we have.
Yes, madam. Yes.
నేను వచ్చాను.
Sir, shall we start, sir?
Okay, Napa, sure.
Madam, good morning, madam. Welcome to Dr. YSR Horticulture University, Venkataramanagudam, West Godavari District, Andhra Pradesh, in Global Connect series, in collaboration with Delaware State University, Dover, USA. The topic is searching for heat stress tolerance in blueberry. So this is the topic. And respected Honorable Chairman, Dr. YSR Horticulture University, Honorable Vice Chancellor, Dr. T. Janakiram sir. Really, sir is this is the program of Sir Brainchild, and this is the 6th Global Connect series by involving all the eminent scientists throughout the world, and to educate our students, scientists, teachers, and all the in all colleges of horticulture, in all polytechnic colleges, all KVKs, all research stations, and all our affiliated colleges also. And madam, welcome Dr. Kalpalata Millimi, welcome madam, guest speaker, today guest speaker. And madam is working as associate professor, plant breeding and molecular genetics, Department of Agriculture and Natural Resources, Delaware State University, Dover, USA. And respected all university officers, Director of Research, Dr. Naram Naidu Garu, Director of Extension, Dr. B. Srinivasulu Garu, Dean of Horticulture, Dr. Padmavatam Garu, Registrar, Dr. Gopal Garu, Dean of PG Studies and Librarian, Dr. Uma Jyoti Garu, Dean of Student Affairs, Dr. Salome Sunata Garu, Controller of Exams, Dr. Venkateswamy Garu, Estate Officer, Sri Mallikarjun Rao Garu, Controller Shashikala Madam Garu, and Rapporteurs, Dr. Sudhavani, Associate Professor, Dr. Vijay, Associate Professor, College of Horticulture, and all the associate deans. Welcome to all associate deans, research station heads, KVKs heads, and polytechnic colleges principals, and all the ladies and gentlemen. Really, it is very happy to share that after joining our Honorable Vice Chancellor, Sir, Dr. T. Janakiram, Sir, really we have already signed almost 78 MOUs, and a great achievement for by connecting all national, international, and private and public institutions by MOUs. And this is the 6th the Global Connect series. and 5 already completed. Every 1 month, we are conducting one Global Connect series, and by which all our scientists, students are knowing the international around what is going on internationally. And welcome, sir. Good morning to all. And so, Dr. Our Honorable Vice Chancellor, Sir Chairman, having some other assignment, sorry, I think may not participate. And sir, before going to the topic, I will introduce our guest speaker, Madam Dr. Kalpalata Madam, who is Associate Professor at Delaware State University, and Madam is University, United States of America. Her research utilizes genomics, genetics, and molecular biology tools. Her current projects focus on improving blueberry, strawberry, hemp, and sugar maple for biotic and abiotic stresses. She obtained her PhD from Oklahoma State University, had postdoctoral training at the University of Northern Iowa, Iowa State University, and Delaware State University since 2008. She has been working as at Delaware State University in various capacities. Dr. Melme is originally from a small village called Erlampalli, Chittoor district of Andhra Pradesh. Madam is our Telugu madam, and attended government Telugu medium schools and junior colleges in village. She obtained her BSc in agriculture from SV Agricultural College, Tirupati, and MSc in plant breeding from the Faculty of Agriculture, Annamalai University, Chidambaram, Tamil Nadu. After her master's degree, she worked as a research associate for 3 years in ICRISAT, Hyderabad, and briefly as agriculture officer before joining her PhD at Oklahoma State University. Dr. Kalpalata Madam is married to Dr. Sacha, who is also a professor at Delaware State University, and have 2 daughters in college. And Madam, at almost she is handling at Delaware State University nearly 20 lakh US dollars projects, almost projects she is handling, various projects to read. So much biodata is also there. Madam is already teaching for BSc, MSc agriculture students, and her at credit 24 national and international publications published in various journals of international reputance, and is guiding MSc students, so many students she is guiding, and also acting as member for the many students at Delaware State Agriculture University. And Madam credit awarded almost 10 awards at international levels, and the latest award is Travel Award by OSU Plant Bionet for research presentation at the Plant and Animal Genome. 15th conference, San Diego, California. So to read so many awards and scholarships, she got almost 10 awards. The Madam has already got, and academic qualifications, state representative for the state of Delaware in the National Plant Breeding Coordinating Committee, and Madam also professionally affiliations with the American Society for Agriculture Science, National Plant Breeders Association, Crop Science Society of America, American Association for Advancement of Science. So, credit nationally and internationally reputed work she is handling there. And welcome, Madam, and good morning. Thank you for accepting this invitation and to give the lecture on searching for heat stress tolerance in blueberry. Welcome, Madam.
Thank you very much, Dr. Sanjay Rao and Registrar for inviting me for this presentation. I feel honored, and you gave me an elaborate bio introduction. So I think I can shorten my introduction with my slides. Thank you very much. Really, I feel honored to be here and share my experience and my research with our students and professionals. I also, since I am in Fulbright, can one of you can take the screenshot and share with me, so that can be a record for my files. Like I mentioned, I work with several different crops. Today, my focus can be on blueberries. So you will see why I have this title. Like for students, so Dr. Dhananjay Rao read so much about myself. So I am not that great person. I'm just a regular Telugu girl born in Chittoor district, went to Telugu medium schools. When growing up, there was no big ambitions. Just wanted to get, my father had ambition for us to graduate 10th grade, have a teacher's training for 1 year and be a teacher. That was the humble goal they had it. But you know, when you put your heart and doing something, and you will reach beyond your expectations. So since my dad was a teacher in the high school, I just wanted to be first in the school, in the class. So that gave the attention when we were in the intermediate to our lectures. They suggested that we can write entrance exam and go to so and so. So one lead to the other. So that's why I am here now. And I'm really satisfied with my journey. So there is no regrets. So as I'm coming from Chittoor, so you can see a mango orchard and bananas. After my 12th grade in Airala, BSc Agriculture, I studied in BSc Agri in Tirupati, and then Masters in Annamalai University in Genetics and Plant Breeding, and Oklahoma for PhD. So in meanwhile, I worked.
Madam, please unmute, madam.
Madam, unmute, madam.
You should have warned me. So sorry, guys. Okay, sorry. So I really feel honored to be here today and share my experience with all of you. Thank you for inviting me to this presentation and to be part of this global series talks. And Dr. Dhananjay Rao gave me an elaborated bio of my introduction. So I will skip some of the things that I prepared because we don't want to board with my self dabba. So just I can skip. Like I mentioned, so I grew up in a village in Chittoor, in a farming family, and there were no big ambitions when growing up. Just wanted to graduate from 10th grade and had a 1-year teacher's training. So that is the goal my father and my grandparents had, at least for us. But when you put your heart in something and you can achieve beyond your expectations. So even though we were in Telugu medium schools, since my father was a teacher there, or at least in high school level, my goal was to be first in the class. So that led the recognition from my teachers. So they suggested when we were in 11th and 12th grade that we can write entrance exam. Until 12th grade, I had no idea that there is an entrance exam for medical school or agriculture and so on. So after 12th grade or intermediate in Airala, so with the entrance exam, then I joined the agriculture college for BSc in Tirupati, and then in Annamalai University for masters, and then Oklahoma State University for PhD. After my masters, briefly I worked for 3 years in ICSAT and also in department agriculture officer. After having married and 2 daughters, then we decided, both me and my husband decided to take a PhD, study PhD program. So that's where we joined Oklahoma State University. So this is my family, current family, nuclear family. Though we have extended family, so my husband, myself, and two daughters. So they both are in the college. And my other daughter, Pluto, her name is Pluto. So now we can come to the topic. So feel free to ask any questions in between. So like in my bio, I worked with so many crops from sorghum, rice to pearl millet to beans, so many crops. But in 2016, I was appointed for the small fruits breeding program in Delaware State University because we didn't have a breeding program in the state, state breeder or breeding program in the state. Though we have strawberries, blueberries, and other small fruits growing in this region. So then I thought about what projects I started these projects from the scratch. So there were no resources available before in the university for me to head start. So blueberries and the strawberries got my attention. The reason is because now the blueberries are enjoying the status of superfood with having a lot of antioxidants and providing health benefits to the human. So and so we can talk about. So that, and also I was looking for where I don't want to be competing with someone already established programs. So I just wanted to make my signature in the world with my own programs. So that's how I started this one. And looked at the different scientific problems that are available. So definitely blueberries are a superfood, and with the statistics that you look at it, they have expanded substantially last 15 years or even 1.5 to 2 decades. I will talk about it in the next slide. So major focus is having it's a superfood. People enjoy it, and it's worth studying about it. Also, blueberries are a very young plant, so our young crop, I can say. So meaning that they have been recently domesticated. So the domestication happened for blueberries only in early 1900s, so 1910 or 1920 that time. So before that, they were grown only in the wild. American Indians. So those are native people in America before European immigrants. They use blueberries and cranberries and several other berries for their cuisines. And then they also use blueberries, blackberries, and raspberries for various medicinal purposes also. Like how we use like garlic and ginger for when our cold cough. So they use these berries for different purposes. If you have a fever, you will use some different type of berries. So that's what they were doing. And in 1900s, 1910 or 1915, there was a scientist in New Jersey, and under his assistant, they thought, why can't we grow blueberries as a commercial crop? So why can't we establish, why can't we develop varieties from those? So they went on the expeditions and collected the best bushes that they can think about and planted them and started selections and released varieties. So from then, blueberry industry started growing. So when the plants has been grown commercially and industry also established. Even until 1900s, 1990s or later century, the geographical region that grows blueberries are mostly in northeastern United States. But now, because of the health benefits and the establishment of industry, The blueberries are grown in many states in the US. You will see in the coming slides, and also in many countries, including China. China is vastly, I think it expanded its blueberry acreage more than tenfold in last 15 years. So it is becoming an economically important crop in many countries, especially in cold where you have the cooler temperatures. So when the industry is established, when you have demand for the crop, you also will have more problems. And also as a plant breeder and plant scientist, we wanted to tackle those problems, as well as we always strive to provide better products, right? So better products meaning in the blueberries in terms of the nutritional quality, adaptability, also the climate readiness, the crops that can be climate readiness. So this is where my topic for today comes under.
All right, but first you have to unlock your device.
So just to give you a brief idea about blueberries, it's not just one species, blueberries. It can be around 200, somewhere from 200 to 400 different species comes under a blueberry category. And again, within that, you'll have a northern high bush. So this is the northern high bush. And also you have southern high bush. And then you have a rabbit eye. These are different types of blueberries, depending upon where they can be grown. So I'm explaining this so that you will have an idea about how different crops, even though we may have a same name, but you have different types within the crop. And then the different species will have a different traits to contribute. So, like I mentioned, even now, the wild berries are still famous. Most of the frozen blueberries, excuse me. So most of the frozen blueberries and that what you see on the ice creams or cookies and processed foods are mostly coming from the low bush type. Those are grown just grown widely or collected. And high bush, or most of the high bush are cultivated type because they are taller in nature. So that way it's easy for cultivation, harvesting, and so on. So the northern high bush comes where, let me put my laser pointer, sorry. So you can see my screen, right?
Yes.
Okay, so northern type, you have polar regions, and these northern type requires at least minimum of 700 hours of chill hours, meaning less than 7 degrees centigrade. So in order for the plant to vernalize properly and produce fruits in the coming summer. So that is the chill requirement for northern high bush. For that reason, they were mostly grown in northeastern part of the United States. And the southern high bush type are grown in Georgia, North Carolina, a little bit southern type. So southern type requires minimum of 400 hours of chill hours. So southern types are basically derived from northern, majority of the genetic background is from northern type, except that they have been crossed with one species, it's Daraway, that is now mostly grown in Florida in the subtropical regions. So that contributes for adaptation to the warmer region, little bit warmer region, but the fruit quality and the horticultural traits of the crop is desirable from the northern high bush. So that's why southern high bush is derived mostly from northern high bush, but the 90% of the almost 90% of the genetic background is from northern high bush. 10% or less maybe from the another species called Daraway. So you will learn about that in the coming slides. So you are with me so far, right?
Yes, madam. Yes, madam.
Okay. So now, originally, Michigan, New Jersey is a top producer at some point of time. The origin of blueberry industry also started from New Jersey. But now the industry has been expanded so much now that Georgia and California are the top producers. So in California, a lot of blueberry plantations are there. So you don't have the similar environment where you have here. And then Oregon and Washington also has a blueberry, substantial amount of blueberry industry. So we know why the northern high bush type are grown in colder regions, then why I'm studying about heat stress, right? That is the question. So when I'm trying to find the project start, what projects I should start, then I see, I notice so many times in the news that the temperature, heat waves that have, heat waves are occurring in the regions that not historically be noticed. So for example, even last year, we had in France, 45 degrees for 3 days in a row. And that was the first time that they have noticed. Same way in Delaware and New Jersey, those regions. Please mute if you're not talking. So that those regions also have a heat waves, unexpected heat waves, and those heat waves stays longer. And usually these heat waves comes in the summertime, June and July. So June and July is supposed to be warmer, but not the extreme temperatures. So the extreme, when you have high temperatures of heat waves for longer time, what happens? The fruit quality is reduced. So that was apparent. That was apparent from the field point of view. And then I was also looking at the studies. Anybody has done any studies? So when I started the project in 2017, there was only one study was there that in China that they compared different types that I mentioned, northern type, southern type, and rabbit type, different types for their heat tolerance. And if they can exhibit any difference in heat, any response to the heat stress. So that was the only study that was there. There was no genetic studies or genome studies available for heat tolerance or heat stress in blueberries. So that that excited me. So to start a new projects in the new direction that. So that's how I started my project. So let's go from there. So for students, I don't need to explain too much about what happens when you have very high temperatures. I think we have a lot more students also in this, right?
Yes, madam.
Yeah. So when you have high temperature, like high intensity, high light and high temperatures, so the photosynthetic synthetic apparatus are the first things that that get affected, right? With the excitement of our electrons, and then subsequently the proteins and lipid molecules can be destroyed, and then the water membrane, the cell membrane can lose its function, thereby its water loss, so on. So when you have prolonged exposure to high temperatures, the visible symptoms that you see can be varied from stunted growth to burnt leaves, what we see here on the side, and fruit drop. So that way you have reduced heat and quality. So in very rare cases, when you have a susceptible genotype, death of the plants can occur. These are some of the typical symptoms that you can see on the fruits when you have high temperatures, like above 35 degrees or so. This is the typical symptoms that you see in not high bush type. So, like in my introductory slides for blueberries, I was talking about northern high bush and southern high bush type, right? So northern high bush type, let me minus this one. Mostly derived from Corymbosum. Its genus name is Vaccinium. The species is Corymbosum. So you'll see more broader leaves, and they are deciduous type. So the meaning that in the winter time it sheds its leaves, and you will just see only sticks standing in the plant. And then when probably somebody needs to turn off phone or something, like I'm hearing echo. The other type, the Daroway, is the evergreen type, and most this species can be found in Florida and subtropical regions, so the warmer regions, and the southern high bush type are derived by crossing these two. And again, I forgot to mention ploidy levels. If we talk about the ploidy levels, in blueberry, you have diploid, tetraploids, pentaploids, and hexaploids. So various types of ploidy levels are there. So in general, most of the cultivated, commercially cultivated blueberries are tetraploids. So we want bigger plants, bigger seeds, so on, so right. And there are some off-height bush type. So those are pentaploids, crosses between there are so many other species also. So derived from interspecific crosses. But I started working with diploids. So what you see in the slide are both of them are diploids. So the reason for, since I wanted to work with the transcriptomes and genome, so I don't need to deal with multiple copies of the same gene. So that's the reason I started with diploids. The advantage, one advantage is easy to deal with assembly, and then not having to deal with multiple copies of the same gene. The other advantage is the Daroway is very compatible for crossing. So if if I find any traits that are interesting, I can transfer those traits to other species using the Daroway. So today's my talk, I will divide into two. I will talk briefly about two projects. One is with the transcriptome analysis for high temperature stress, and then other one is with Jivas. And then at the end, I can talk about how we are merging both of them to find the candidate genes, right? So that way it will be useful to see the end product. So for transcriptome analysis, I chose these two divergent plants. Corymbosum is the northern high bush, adapted to the cooler climates, and Daroway is the species Daroway is grown in subtropical regions, adapted to warmer climate. We made a note of the morphological traits. Morphological traits, I will talk about it. And then we want the experiment to be as natural as possible, that what we see, if the plants are left in the field, how they can be exposed to the environmental parameters. So first we let the plants at 25 degrees for adaptation for a few hours, and then started the heat stress. So the heat, the temperature that we imposed for transcriptome analysis is 45 degrees. The reason is that this is the temperature that in the previous study, they were, when they looked on the microscope, they can see the difference in the chloroplast response, the destruction of chloroplast in sensitive varieties versus the resistant varieties. I thought that may be a good idea for me also to see, to impose that temperature for transcriptome analysis, right? So when you have a 24 degree, when the ambient temperature is there, you don't want to suddenly just put 45 degrees. So you have to increase gradually, mimicking the day temperature that we see, right? Morning, when the sunrise comes, you have a lower temperature, and slowly you have higher temperatures. Same way, the gradient was applied. So once the temperature reaches at 45 degrees, so we collected samples at 0 hours, 6 hours, and 9 hours. So again, these are arbitrary. We may, in the next experiment, we may conduct at a different hours. We don't know. This is our first experiment, so to try. So these are hours that we chose based on the image analysis. Image analysis that we, I think one of my students, she also conducted a trial experiment to decide what hours are best for us to choose to collect samples. All right. So once we have the experiment done and the samples are collected, then next step is for transcriptome analysis to isolate RNA and do the quality check, prepare libraries, and then sequencing. So once we have this all, the library sequencing is done by my collaborators. I will explain you in the last slide. And then analysis. After the quality check, trimming, and so on. So once we have the good quality feed, then we do the analysis by finding differentially expressed genes, and then also using I think someone needs to be turned off their phone. If you have, you might have logged in with two accounts, and I can hear your phone. You can mute, yeah, probably mute and see.
Hello.
Yeah, so
Please mute.
Yeah,
okay, madam.
Yeah, I'm hearing echo.
Again.
So yeah, I think it's okay now.
Okay.
So please ask questions as we are discussing. So these are just to give you a morphological difference. So these are Corymbosum is more broader, comparatively broader leaves. These both are tetraploid here. What you are seeing is tetraploid. In the previous slide, what I shown are diploids of the same species. So these are glossy, and they have the Darovoi has a slightly girophytic characters, like waxy coating, and then a small, little bit thick leaves. The orientation of the leaves are slightly different from the parallel orientation here than the Corymbosum, where you have angular orientation in the Corymbosum. I think that traits also helps for the Darovoi to adapt in the warmer regions. And like I said, people prefer to have blueberries like this. The bush and the fruits, taste, everything is likable horticultural traits are desirable from the Quorimbosa. That doesn't sorry, that doesn't mean that the fruit traits in Daraway is not good. We can, that's a different topic. We can talk about it some other time. So we covered this. So when you wanted to find differentially expressed genes or candidate genes, it is also do the transcriptome analysis. It is also very important that those plants are exposed to the environment that you are looking for, right? So we wanted to make sure, we wanted to see morphologically, are there any difference in terms of response of these two species for the high temperature stress. So the leaf was imaged using the laser scanning confocal microscopy. So you can see here, this is organelles inside the stomata. So in the stomata, you have a guard cells, right? In the guard cell, again, you have chloroplast and other organelles. So these organelles were autofluoresced with red dye, and then we looked at it. So when you see in Quorimbosa, you don't see a big difference in the size of the stomata or the organelles. So, sorry, I'm getting the cough because the round here, what you see is based on the organelle fluorescence. So whereas in the Daraway, you can see by 9 hours, you have a reduced size of stomata than the stomatal opening than the 0 hours. Yeah. So so this is again, I don't have a picture to explain here. But what we see here, some of the intense red dye is from the chloroplast also. So chloroplast thylakoids. So that was I don't have the picture for that one, but we had the measurements taken using the G-stack mechanism also. What my student observed for those in Daraway, the thylakoids stayed intact. Like how if we put all the coins in a small tiny purse with a well-organized fashion, so you can put more coins in the layers and just keep it tight. But if you just unzip that purse and then coin becomes not organized, they will be spilled all over the place. So she observed that in Quorimbosa, the thylakoids were disrupted. They were not organized as we see in the pictures. Whereas you can see the same pattern of tightly organized thylakoid layers in Daraway. So probably in some, I don't want to overstate, but there is some other mechanism that Daraway is coping up, not to destroy the membranes and so on. So that way the thylakoid stayed intact and the stomata can respond as normally they can respond to any heat stress. So whereas that mechanism failed in Quorimbosa, maybe that's why it is more thanks to. So just this is a theory. So no proven experiments done yet. In little bit lesser zoom level, you can see the pore size of the stomatal pore size in Quorimbosa. So you don't see a big difference from 0 hours to 9 hours. They're all the same magnitude. And here in Daraway, you can see the difference between 0 hours to 9 hours. So where the 9 hours, the opening is it's very obvious that we can see the difference. Just give me 5 minutes, I'll be back. Just check if my seminar is started or not. Okay?
Okay, madam.
Thank you very much. Just give me five minutes, I'll be back. Okay.
All are requested to stay for 5 minutes.
Not too much, half cup is fine. Sorry for the interruption.
Okay.
So after we confirm that the experiments that we imposed are really working and the plants are responding differentially, so we went ahead and sequenced the RNA sequences. So this is a big table. You don't need to be overwhelmed by looking at it. I just wanted to show you here few things. So to tell the coverage, so we had a good coverage for the genome. And the length of the contigs, the length of the gene, the assembled genes or transcripts are reasonable. The number of transcripts and the length of the contigs. So this is just to have an idea, overall idea about how many genes within the differentially expressed genes, how many genes are upregulated and how many genes are different downregulated. By looking at this graph, you really don't get a big information out of it, but it gives you an overall idea, the trend, how it is going. So here you can see in corimbosome, what these are compared with 0 hours. So 6 hours at 40 degrees, 45 degrees treatment versus 0 hours, 9 hours. So that's how we compare. And you can see in both the cases, you have more number of upregulated genes. And in the Daro way, you have 6 hours, you have more number of upregulated genes compared to downregulated ones. 9 hours, we have a lot more genes downregulated. So like I was mentioning in my PhD, I had a contrast experiment done. I had 2 genotypes, Firmicutes, cold tolerance, cold susceptible, and I exposed them to cold. If we look at those graphs, the experiment done for cold tolerance, so how I have done for the heat size, same trend I can see here. My susceptible, so the tolerant one has a lot more down-regulated genes as the plant gets adapted. Our plants, the treatment increases, the number of hours for the treatment increases. So one reason I assume is that
ఫోన్ లో అదే తిరువూరు బస్ స్టాండ్ దగ్గర
Yeah, please mute.
Please mute.
Not talking, please mute. Thank you. So my assumption, or not only my assumption, in many papers that you see, if you have more downregulated, the tolerant one might be adapting to the conditions that we provide by shutting down majority of the genes that are not required for maintenance. Only genes for that are active or regular maintenance and survival. So that's why you have upregulated genes. I was really surprised and also excited to see this graph by comparing with my previous studies. This is common Venn diagram that we see. We wanted to see out of all these differentially expressive genes, which are common between the species, what are common between the treatments, so on. So in overall, we had 94 genes that are common in all different species and different time points. So depending upon the use, so you can, depending upon the purpose, so you can pick and choose what genes you want to work with. So for now, I am sticking with the 94 common genes because they are there for both the species and on the time points. So that's how I got interested. So maybe in the next time, for the next experiment, I may be interested to look at a different set of genes for a different purpose. So for the coming slides, I'll be talking about these common genes. Right? So now you have the differentially expressive genes, and then what we can get out of those. Right? Main interest was to see what kind of functions they are involved. We don't have annotation for a lot of them. Like in Arabidopsis or for example, the majority of the genes has been characterized. So, but still, we can get a broader view of which pathways they are involved in. So here, the bigger the dots, meaning the more number of genes. So in the x-axis, we have genes, and these are the different metabolism, the different pathways involved in that. So when we see there are a good number of genes in all four different columns involved in metabolic pathways. There are also a good number of genes involved in thiamine and purine metabolism. You can see across. And this is just too much overwhelming, but major is we can look at what the focus can be the major pathways that are involved. The bigger the bar, more number of genes involved in that particular pathway. So we paid attention only for those major genes involved in major pathways, or more number of genes involved in whatever pathway. So that is the major focus for this project. So when we compare, when we look at the pathways, individual pathways that are involved, so we can see a lot of them are involved in endoplasmic reticulum. I think in the previous slides also, we can see, so there were a lot of them are involved in endoplasmic reticulum and ribosomal pathways. So either endoplasmic reticulum, ribosomal pathways, so they're all involved in what for students? Protein synthesis, right? So there was a lot of protein degradation is happening, new proteins are synthesizing. So we can see that is the reason we can see many of the differentially expressed genes are involved in this ER pathways, endoplasmic reticulum, or in ribosomal pathways. And there are slight difference between Darway and Darway and corymbosom. So how many number of genes are upregulated in these pathways? But we can talk about some other time. So that gets too much detailed. And fatty acid degradation. So fatty acid degradation is one of also mostly this activity associated with any abiotic stress responses. And this is just validation for RT-PCR. Then for this project, any further details if you are interested. So we have the paper published. So please go and visit. You can find more details, or you have my number, so you can contact anytime. So that's one project. So that project mostly done by my master student. And then, so now we have the pathways, right? So sorry, genes, transcriptome, at least DEGs that are from the transcriptome analysis. The next goal for us as a plant breeder, we wanted to look for associations, right? What genes are associated, or QTLs are associated for this trait. So that is our next goal. In this project, I will briefly explain that. Sorry, I'm having a little bit of tea also. So in this project, we did GBS using by the population that developed from the Darway and corymbosom, with taking the advantage of Illumina sequencing. The phenotype for the high temperature stress for the F2 population, and then association study, identify the SNP, and then identify the genes. And then we also merged the two projects, results from the two projects. I think someone has muted.
Please mute Ayappan garu. Hello.
Okay, thank you. So for this project, we had the population. I started in 2017, and for blueberries to see fruits and collect fruits, it will take a minimum of 3 years. So that means I have to wait another 3-4 years to do my GWAS analysis. So then I looked around and contacted Dr. Nick Borsa. He developed, fortunately, he already had F2 population from these two species, the Darway and corymbosom. Since they are highly self-incompatible, it is hard to have, sorry, let me put my, because of the change of weather, my throat also is giving me a little bit of.
No problem. No problem.
The change of weather and moving to so many places. So we have the OPB. OPB is both of OPB are corymbosom, and the NJ88 New Jersey lines, these are Darway. And then he made classes and has F1, but we cannot, unlike in rice or soybean or other beans or any crop, we cannot make a F2 by helping the F1. The reason is self-incompatibility. So we have to make a pseudo F2s by crossing two F1 population. We had a few clones in this one and few clones in this one. So around 1,500 plants we had in the F2 with the reciprocal crosses. That's a good number of plants, but we work with around 300 or 320 plants for this study. So for all those plants, the F2 population, parents, F1, and then also I had some cultivars. So including all of them, we had GBS done for this population. The most of the read length is this one. And major take-home message from this slide is 85, so almost 85% of the sequences had a mapping, had matched to the genome sequence, blueberry genome sequence. So that's a good thing. And one must be wondering why these numbers are not here. These are chromosome numbers, but when we started this analysis, we didn't have a deployed sequence, deployed genome sequence. So these are scaffolds from the tetraploid, the longest for any homologous chromosome. So we took the longest sequence. That is why you don't see the numbers in 1 to 12. So you have different numbers, but it should not be a matter. So GBS is libraries are prepared with one enzyme. So some people use 2 enzymes to chop the DNA, but we used one FK enzyme. I think that also gave us a good number. And finally, we have our filtered good quality SNPs, 126,000 good SNPs that we can use it for a downstream analysis. So which comes under the frequency about one SNP for every 250 base pairs of genome sequence. So that is a pretty good sequence, the frequency. So that the last 2 slides, we looked at the genome analysis and SNP delivery, development of SNPs. And here in the side by side, we had heat stress experiments conducted 2 years, and actually 3rd year also, but in the 3rd year, we had a reduced number of plants. So for 2 years, we had all 260 to 300 plants, F2 plants, along with the F1s, parents, and so on. So the data collected for NDVI, greenness index, quantum yield, and chlorophyll fluorescence related parameters. So these are the parameters data were collected. You can see this is the young plants, 1 year old, and then you can see here these are 2.5 year old plants. So these are all these plants. What you see, this is after they expose them to heat stress. So you can see some of them are susceptible wherever you see the scorching, and some of them are good. Because here we expose only 40 degrees centigrade daytime and nighttime for 3 days. The reason is we don't want to lose plants. The F2 plants, it's hard to derive, right? So we don't want to lose them. That's the reason we change our condition, the treatment conditions. Some examples here. So the top rows, they stayed green even in 3 days at 40 degrees. Here you can see some of the scorching and everything going on. But these are all the F2 plants. That means we can see the differential response in F2, so which is a good thing. So we can also have traits of we have a chance to select traits of our interest. Then you also need to know whether these genes are quantitative, whether they are quantitative or Mendelian genes, right? Mendelian traits. So for most of the parameters that we measured, we can see approximately bell-shaped curve with the frequency distribution. And the box plots can tell you about the range. So this is 2019 data range, and 2020 is for the quantum yield. And these parameters or analysis is to give you a gauge of the data. So fluoropen and polypen, these are the two handheld devices utilized for measuring these traits, and this can be a non-destructive type. So you don't need to sacrifice any plants for taking the data. This fluoropen gives you chlorophyll related, photosynthesis related parameters. Polypen will give you greenness index, NDVI, so and so. Very simple to use. So now you have the SNP information matched to your genomic sequences. And then also now you have the traits, right? You have the traits measured. So when we do the Manhattan plots, we can find some associations. So yeah, you would like to see, sorry, you would like to see SNPs that have been associated for more traits. So that would be more easier, more desirable, we can say. So here you can see these SNPs, and this one as I had to look for that. I couldn't see similar. So some of like same. So these are, you see these dots, and this one has more than one trait associated for those. So those can be interesting SNPs. So basically, we wanted to see with the high threshold and also a SNP that is associated with more than one trait. So with this analysis, we have around 245 SNPs that are significantly associated with more than one chlorophyll fluorescence parameter. And when we do the blast search analysis, out of 245, 179 SNPs were present in the genic region. So that means the possibility of finding candidate genes is higher, right? So only 26 were in the intergenic regions. Like in transcriptome analysis, we have more heat shock proteins, and here also we have more of molecular chaperone proteins involved in this one. So you can see the parallel results between these two experiments. It's interesting to see, since we have the transcriptome, and out of from the Jivas, we have around 176 genes that are in the that are present in genic region. So we wanted to see if what percentage of genes or what genes are common in both the experiments. That may be a more reliable source, right? Those can be a good candidates to select for further like molecular breeding selection or creating selection models, any of those. So that's where we compared both of them and selected some candidate genes from both. And I think one of my staff member now, he's interested to validate this information, whatever the results that we derived using the RT-PCR for the GWAS data. So the GWAS also we use BLUF analysis to pick up the candidate genes. So to summarize, so we can, like in the pictures that we saw, top rows and the bottom row, so we do see significant variation among the different plants. responded to high temperature stress. So that's a good thing. And then we also have the traits that we measured and do distribute in a quantitative gene fashion. So that is amenable for the studies. GBS protocol, the GBS, we used only one FK enzyme for chopping the DNA. So that also gave us the successful results. But some people also use in combination with other enzymes as well. So GWAS identified significant associated SNPs. And with these SNPs, we also find some candidate genes. And we are cross-validating these candidates with transcriptome data. From this finding, one next level, what we are doing is this year, we are again having, instead of all 300 plants, we'll have 100 F2 plants. And then we can do test these candidate genes and also develop a model. And then from this, we can extrapolate to the other F2 population or other population to find traits or genes associated for the heat stress tolerance. So that is where I am. We are in the lab with this project. I will be happy to take any questions. So before that, I can also introduce my lab members. Krishna is the main person for the GWAS in this project who did the GWAS experiment. He's a postdoctoral scientist. And then for the transcriptome analysis, we had Jody, master student, and she's now at Iowa State doing her PhD. And Amar is also a postdoc. Now he is in India, coming back again, joining the lab. He works on strawberries and maple. Raslin works on strawberry master's program. She graduated. Aisha on strawberries. She also graduated. Lungove or Lulu, she worked on sugar maple transcriptome for drought stress. She also graduated and she's a teacher now. Byron Manjanaro, he's from Belize. He worked with blueberries. Looking at the genomic relations between different species of blueberries. So main goal is to find what is the difference at the genome level from the wild species versus cultivated species. So he graduated with master's in my lab, and now is a student, a PhD student at University of Florida. Aditi is undergrad. She was helping grad students. Heyman is working on strawberry anthracnose, a master student currently. This is myself in Orlando. That is supposed to be oldest plant tree or something. So I was visiting and took a picture from there. So I think, and then I have my collaborators. So Dr. Nick Orsa is a plant breeder. He has experience in blueberries and cranberries for more than 35 years. So released so many varieties in blueberries and cranberries. He still enjoys royalties from those varieties. And Dr. Umesh Reddy is one of my friend and colleague who is the main contributor for DNA sequencing and analysis for us. Of course, without funding and the money, we cannot do all this research. And most of my support is from the United States Department of Agriculture. So with that, I'm happy to take any questions if you have.
Thank you, madam. Thank you very much for a wonderful talk on blueberries. And that actually, blueberry is you stressed on the qualities and advantages of what are the nutritional point of antioxidants, reduce heart attacks, and blueberry. There are a lot of industries going on at the USA, and the effects of high temperature on blueberries and the two species, Vaccinia carimbosa, Vaccinia derovi, and what are the stomatal variations in when the plants are treated with different temperatures, and how the genes are responded to heat stress. And you also stressed on the gene characterization and evolution of different varieties and genomic sequence traits measuring, and how the sequencing of RNA, DNA is there when they're exposed to heat. So at the molecular level, at the genetical level, you have given a good talk on blueberries, madam. Thank you very much. Now it is the time for question and answers.
Yes, yes.
Please ask all are requested to ask question and answers.
Madam, good morning, madam.
Very good morning.
Very interesting topic, very useful for the students and scientists also. Then especially, madam, that for your first slide is very impressive, madam. Especially the decorative of the banana and blueberry. Yeah,
Okay.
really a good idea. Really, immediately model. Regarding madam, only just by hearing your lecture, this heat stress tolerance genes any relationship with the disease resistance, madam? Have you observed any such data or information?
Yeah, I think there may be. I didn't pay full attention for this. I haven't looked. So there should be common, like some of the chaperones, genes, and no, I don't want to tell whatever. I didn't look at in that direction. No.
Okay. No, why I'm telling is, madam, especially in the heat stress tolerance, there is a chances of, if we observe, there is a resistance mechanism to other diseases also,
Yes.
especially viral diseases. So if we look into that aspect also, sometimes, madam, there is chances of getting a combined.
Definitely, it may be there. I didn't pay my goal so far, like look at this and then develop a model. So I look for the candidate genes and in those, but I can look at it. I don't have in my brain, in my mind, just to give you an idea. I haven't looked at it.
But thank you, madam. Thank you very much.
Anybody? Many students are also participating, scientists. They can ask questions, madam. University officers, zonal heads.
Madam, Kalpalata Madam Garu, one small, it's not a question. I want to have a clarification. Why this blueberry is distributed only along the coast?
Yeah, so there are other.
Why that adaptation? Is it because of migrants went there and settled and there only it is being grown, or is there any other reason?
Good question. So good question. I had, so first one thing is they also very picky plants. They like acidic soils. Like if anywhere if the soil pH is like above 5.5, they don't survive good. So for that reason, they were in the nearby coastal areas, so where you have acidic soils. But in California, we don't have that acidic soil. So last time, a couple of years ago, before the pandemic, I was attending the NAPP National Plant Breeders Association meeting, and we had a tour to one of the commercial companies. When we entered the company, there was a huge tank on there. So that tank was sulfur, actually. So they are injecting sulfur in the water through the water, like fertigation, to artificially lower the soil pH. So, and some commercial plantations, they grow them in the container plots. rather than in the, that's much easier to maintain soil pH than the whole soil. So that can be one reason. And then now I have pictures only where the major states are growing, but in reality, other states are also, almost 36 or 38 states in the US grow blueberries at commercial scale, but not as much as Georgia, California, New Jersey, Maine, we grow. Florida, nowadays we have Florida blueberries also. But from Florida, mostly rabbit eye type come. And even though I highlighted, since I work with the blueberries, definitely they are a good healthy crop and all those, but our jamuns also are equally
Yes.
plants. So,
Yes, yes,
yeah,
yes.
so we have, they also have equal amount of antioxidants and all those.
Madam, here you selected only these two, no, Carimbosum and Darwai. Why? Are there any other varieties, or
Yes.
only because to study this classical, to do this classical work, you
Yeah.
selected only these two?
Yeah. So
Because it is a starting, probably you selected these two.
yeah, so there are different species also there, but the most of the northern high bush and southern high bush has a Carimbosum background. So now many cultivars, the cultivars comes under northern high bush. They have almost 99% is Carimbosum background. Wherein the southern high bush type of cultivars, how many have been released so far? They have minimum of 90% Carimbosum and 10% from Darway, or mostly from Darway, if not from one or two other species. Very few percentage for that reason.
Okay, thank you.
Good morning, madam. This is Dr. Salomi. Very informative transcriptome analysis study on blueberries with your novel findings and excellent technology utilized. Madam, like this stress tolerance biochemically is mostly related to proline. Proline gets triggered under stress conditions. So the genes you have studied are tracked, upregulated during this, madam. Did you find any study
Yes.
on that?
Yeah.
You have showed, of course, several number of genes, and are they among them? I would like to know.
Yes. So you are right. So prolines are one of the indicators for high drought or high temperature tolerance. In fact, I do have the biochemical spectrophotometric analysis for proline for small quantity of the species plants, not for all 300 plants. I do have some genes that are upregulated related to protein. Yeah, you are right.
Okay, ma'am. Thank you. Similar studies are on here also. Thank you.
Okay. In fact, I have another project in a sugar maple. Sugar maple is a big tree, like honey. So they have in US, they use sugar, in Canada and US, European countries, they use sugar maple in like those pancakes and everywhere. For anything, they'll have a dip as a sugar maple. So that project also I have. So proline is the stress indicator for us. So my students do the biochemical analysis for proline.
Okay, ma'am. Thank you.
Any questions from zonal heads, associate deans, teachers, scientists, students?
Madam, this is Tanuja Priya.
Hi, Tanuja.
Madam, it was a very excellent presentation, madam. Thank you for the informative presentation also. Madam, I have one doubt. In case of blueberry, you said that the genome sequencing was done, and so the transcriptome was compared with that particular plant. Suppose if we don't have for a plant or any perennial plant for the comparison with the genome sequencing, which model plant we have to take, whether we have to be we have to take only the perennial plant, or can we go even with the Arabidopsis, which is the model plant, madam?
Yeah, so best choice is go find the closest relative. For me, if possible. If not possible, Arabidopsis, because again, now with the bioinformatic tools, now you can cross compare with any sequence, right? So for my blueberry, we didn't have a genome sequence available in the beginning. So I used Arabidopsis and grapes, grape sequence for comparison. So and then when I had the blueberry genome available and I compared, the top genes, anyhow, they stayed same. I didn't see big difference. Probably if we have more genes have been annotated, more genes, the gene functions have been discovered, then for other crops, maybe some advantage will be there. But now my suggestion is go with whatever closest relative. Also, you can compare. Nothing hurts if you compare with the Arabidopsis.
Thank you, ma'am. And regarding heat shock proteins, madam, have you traced any heat shock protein related genes in your study, madam?
So we had a lot of heat shock proteins. I think second question I didn't understand.
Heat shock proteins, madam. Actually, the gene related to heat shock proteins, especially for any stress conditions, we will be concentrating on that. No, madam. Have you traced any candidate gene regarding the heat shock proteins also? Madam?
Yeah, we have top genes. I think top 10 genes are related to heat shock proteins. In the transcriptome, somewhere I think I may have mentioned also. Yeah, but when I'm choosing, so you can see right here, right? So the top genes that I have among the common genes, differential expressive genes, are heat shock proteins.
Yes. Thank you very much, ma'am. I was very much interested because my PhD study, even I have done in transcriptome profiling of pomegranate, madam. With regard to the bacterial blight stress studies. Thank you, madam. Thank you very much, madam. It was very informative.
Anybody?
Madam, good morning, madam. Madam, thank you for your informative presentation on blueberry, madam. I have two doubts, madam. One is whenever the plants are exposed to the high temperature, it significantly increases the oxidative damage. So, have you observed any anti-oxidative genes and oxidative protein repairing genes in your study? That is one question, madam. Another one, is there any differential expression in fatty acid biosynthesis pathway or microbial metabolism, madam? So that we can observe these pathways means it is useful for the disease resistance, lysine biosynthesis pathway or secondary metabolites pathways. Have you observed these two, madam?
Hello, the line is connected. Madam. Madam, connect. Sir, please wait. Madam line is disconnected. Hello. You.
I can hear.
Sir, Madam power is some problem. So Madam is giving answer on phone. You can hear. Madam, please talk.
Yes, so let me know what question you have.
Please repeat the question. Repeat the question.
Ramanandan garu, you ask question once again. Ramanandan garu.
Sir, Ramanandan garu.
Okay, next question అడగమని చెప్పండి.
Next question, sir. Any clarity?
I didn't hear the question, but I can hear you and other sir, but I didn't hear.
Ramanandan garu, are you in online?
Okay, leave. You ask any other students or teachers want to have a clarification.
Yeah. Anybody?
Madam, good morning, madam. Madam,
Yes, good morning.
I have two doubts, madam. One is whenever the plants are exposed to the high temperature, significantly increase the oxidative damage, madam. So any anti-oxidative genes and oxidative protein repairing genes have you identified in your study? Another one, madam, is there any differential expression in fatty acid biosynthesis pathway, that is microbial metabolism pathway or lysine biosynthesis pathway? Another one, madam, this one Vaccinium coriambosum, that is a cold tolerance species. Why did you choose this one for high temperature heat tolerance?
So good question. All of them are good questions. So corimbosum is mostly grown in, we go from backwards to the front. So the corimbosum is mostly grown in cooler regions, but I am trying to see, develop varieties climate ready, right? So this prolonged heat waves, the frequency of heat waves that we are seeing, it is happening before. So even in northeastern US or in our European countries where blueberries are grown mostly, we have heat waves occurring in the recent years. That can affect the fruit quality. So that is the reason we started this project. And the second question, we did see the fatty difference in fatty acid metabolism that I had in one of the slides, but I don't remember looking specifically at the lysine. At this point of time, I didn't pay close attention to those genes. And the third one, the first question that you asked is for the antioxidant genes that are related to antioxidant. Yeah, we do have like some of the proline metabolism genes not in protein metabolism and some other genes, but I don't have the list in my head, the candidate genes that I'm looking for, but we do have in general the genes that help scavenge antioxidants. Sorry, I'm not state to feed apples.
Thank you, madam. Thank you.
I hope it helps now.
Are you clear?
Fine.
So, anybody? Any questions?
Good morning, madam. Hello. Myself Nagalakshmi, madam. Madam, after identifying that gene, heat stress tolerant gene, have you done any transformation to heat susceptible genotypes of blueberry, madam?
Voice not clear. Can you please repeat the question?
Nagalakshmi, repeat it.
Okay, sir. After identifying that heat stress tolerant that gene, have you done any transformation work to heat susceptible genotypes of blueberry, madam?
Madam, after identifying the gene, any transfer to susceptible variety like that she is asking.
Okay. So I don't know whether it can be a one gene or several genes, but when we saw it is a quantity to train. The majority of them are quantitative traits. Right? So with this, I don't have answer exactly I can transfer or not, but transfer in which way? So selecting by selection can be helpful, or by regular I can do the transfer to even the tetraploids utilizing my Darrow-Way plant. So with the Darrow-Way, it forms unreduced gametes. So even though it is a diploid, I can still get tetraploid or a triploid genome.
Nagalakshmi, you got the point?
Oh, okay, sir. Thank you. Okay, madam.
Yeah, because it is not a single gene, it is a quantitative genes, many genes.
Yes,
So like that, madam is telling.
Okay.
So any question? Any questions? Hello.
So if there are no questions,
Yes, sir. Yes, sir.
you come to end.
For more
Yeah.
questions, I invite our registrar, Dr. K. Gopal Garu, to say a vote of thanks.
Yeah, thank you, Dhananjay. First, we thank our Honorable Vice Chancellor, Dr. T. Janakaram sir, for starting this Global Connect series for the benefit of researchers, teachers, and research scholars, and to expose them for international to international experts through this kind of talks. Thank you, sir. Thank you very much. And this is one of the talk series started last year and running successfully, and all the teachers and students getting benefited. And again, this is connecting these young researchers and also research scholars who can think of their research in other countries too, and going doing higher education in foreign countries. And next, I thank Dr. Kalpalata Madam, who is an associate professor at Department of Agriculture and Natural Resources, Delaware State University, USA. Madam is a 1989 batch graduate from SV Agriculture College, Tirupati. So she is alumni of APIU. Really, it is very, very happy for this. Madam, really, your presentation is very nice, and the topic itself, it is very interesting because it is one of the less researched crop and indigenous crop. It is being grown for many centuries. And again, madam, the presentation was very nice, and it is very well explained in very understandable way. So hope all the research scholars, teachers got benefited with this talk. Thank you, madam. Really, it is very interesting talk because we have a lot of young minds in our university, and this is definitely encouraging one for them because this shows a lot of research work can be initiated in indigenous vegetables and also fruits in our country also. And I hope our researchers can have a collaboration with Kalpalata madam through even we can think of MOU and all, and then sure it will be very much useful. And I also thank all the university officers for participating and interacting in this lecture. And thank you, sir. Thank you all by name. Dr. B. Srinivasulu, Director of Extension, Dr. E.S. Padmavathamma, Dean of Horticulture, Dr. Venkataswamy, Control of Examination, and the Director of International Programs, Dr. A. K. Dhananjay Rao, and Salome Sunita, the Dean of Student Affairs, and all the university officers. Thank you for participating and interacting in this lecture. We also thank our rapporteurs for recording the proceedings of this talk. And I also thank all the students, teachers, and other participants who participated and interacted and made it a grand success. Thank you once again for giving me this opportunity. And I also thank once again our Honorable Vice Chancellor who started this Global Connect series. Thank you, sir. Thank you very much.
Thank you, sir. Thank you, madam. Madam, thank you. Thank you, one and all. Thank you.
Thank you, madam. Thank you. Very nice presentation. Really, it is encouraging for to all the youngsters who can work similar kind of things and indigenous fruits and vegetables in India. We have a very, very, very large crops to do this kind of work, and definitely it will be useful, madam, because because of increasing temperature year after year